Thank you all for joining us today. We are going to have our last webinar for Time for Genetics with the topic of Are All Variants of Uncertain Significance Created Equal? A Rough Guide. And we are thankful for the funding that we've received for this program. Mountain States Regional Genetics Network is funded by the Health Resources and Service Administration, or HRSA. You can reference your screen for that funding statement or visit hrsa.gov for more information. I am your host and co-host today with Michaela Welsh. My name is Christy Weiss. I'm the projects manager for Mountain States Regional Genetics Network, and Michaela is our project coordinator. We're here to help with any technical issues, so please seek us out in the chat if you need anything. And just a couple Zoom logistics. If you are joining us today, we ask that you stay in mute during the webinar to preserve the call quality. You can find that here on the mute and unmute button. Please feel free to turn your camera on or off if you're comfortable. We are recording this, though, for archival purposes, so please note that. And then at number 3 here, you can find the active participant list. And then number 4 is the chat feature. We are asking if you would please use the chat if you have questions during the presentation or for our Q&A session afterwards. And then again, if you're having any technical issues, please feel free to message myself, Christy, or Michaela in the chat. So this webinar is part of a program called Time for Genetics. It is a pediatric primary care genetics education initiative that was started in the Mountain States region during this grant cycle. Mountain States is an 8-state region, and you can see on the right-hand side here are 8 states, as well as little pins for all of the cohorts of clinics that have joined us for this education program. We have 28 clinics participating across our region, and we welcome them here today, as well as all of you who have joined them for this informative webinar. The Time for Genetics initiative has explored various topics, for primary care providers in our region, from red flags to genetic testing to autism to seizures, and to this very last topic area, which is variants of uncertain significance. If you're joining us for the first time and you're just now hearing about Time for Genetics and would like to explore some of these topics, this QR code will lead you to this library. That's the same QR code here, or you can just go to mountainstatesgenetics.org/T4G library, and you can access all of that archived information and webinars and topic areas there. We also have some other educational initiatives and information on our website. We have just completed 3 virtual genetics summits, and all of those speakers and those talks are archived on our website as well. This QR code will take you there. You can look at any of those archived topics if you'd like to learn about other various information about genetics. And then I'd like to invite all of you and the families that you serve to join us for our last genetics pop-up coming up on February 22nd. This is an outreach to families and a place that families can come to ask questions about genetics. We will be focusing on the topic of hope through support because it is in honor of Rare Disease Day, which is coming up at the end of February. And then last but not least, just another announcement. We are currently seeking families who have had positive newborn screening outcomes for a listening session coming up in March. If you know of any families in our 8-state region that you would like to pass this information along to and have them participate, this QR code, or you can look in the news section on our website for this information to learn more about this. But we would really appreciate everybody sharing the information with families in our region. And with that, we are offering continuing education credit today for this webinar that is being done in collaboration with Projects and Knowledge. The CME that will be offered are for physicians, MD-DO. We have physician assistant CME and CNE or CE for nurses and CPE for pharmacists available today. A little bit different than in the past. We are only offering live credit for today. So watching the webinar live and then filling out the accreditation form. We will not be offering enduring credit since our grant is wrapping up at the end of May. So please be sure if you are looking for CME for this presentation today that you fill out the evaluation form that will be shared at the end of this webinar. Our speakers today, Dr. Elias discloses that he is a consultant with Illumina and part of their pediatric genetics working group, and Jaclyn does not have any financial relationships to disclose. So with that, it is my great pleasure to introduce our two speakers today, Dr. Elias, who is a clinical geneticist and medical director of the Department of Medical Genetics at Shodair Children's Hospital, along with Jaclyn Haven, who is a certified genetic counselor, chief administrative officer of medical genetics, also from Shodair Children's Hospital. And they will be talking today about Are All Variants of Uncertain Significance Created Equal? A Rough Guide. And with that, I'm going to stop sharing and give them the floor. Thank you both for joining us today.
Thank you. Thank you, Christy. Let me first start here sharing. Make sure I make this correctly. Let's see here. It's the 3 here. Make sure that you, Christy,
That looks great.
Can you see
it
that?
looks perfect.
Great. Okay, let me just make sure I can see the Zoom still that in case there are any. Great. Well, let's get started then. Hello, everyone. So I'm Elias. Jaclyn is here too on the screen. And we both appreciate you all taking time out of your day today to join us here at MSRGN for our Time for Genetics webinar. And as Christy mentioned, our title today is Are All Variants of Uncertain Significance Created Equal? A Rough Guide. So variants of uncertain significance. One may think that it's perhaps not the most popular topic in genetics. After all, who likes to receive a report with a bunch of VUSs? There may be a cause for confusion, dread, really anxiety, dissatisfaction on the ordering provider side, who maybe doesn't know what to tell the patient, on the side of the genetic counselor or the medical geneticist who have to communicate these results perhaps. And then of course, also on the side of the families and the patients who really may not know what to do with these and may not always get the optimal information about those. So, however, I hope that by the end of today's webinar, you'll find that understanding the concept of uncertainty in the interpretation of genetic information brings you right to the heart of genomic medicine. So knowing what all goes into the classification of genetic variants in general and of variants of uncertain significance specifically can help you gauge the relevance and perhaps paradoxically the significance of a particular variant of uncertain significance for your particular patient. And that's really part of the promise of individualized medicine. So we'll divide this a little bit up today. We have 2 parts. The first, probably 2/3, I will give you some background, and then the last third, Jaclyn and I will have a discussion. It'll be more of a discussion about a few different cases, which we hope you find interesting. So here's what would like you to take away from this webinar. First, genetic testing is all but one component of a genetic diagnosis. So what are the other components and how do you integrate these components to synthesize the actual diagnosis? Then variants of uncertain significance represent one category of genetic variants. How are they defined? What are the other variant categories and how do you tell them apart? And then what do you need to know about VUSs before ordering genetic testing? Also, what are some of the aspects of pre-test and post-test genetic counseling related to VUSs? And how do you interpret a genetic test report with a VOS. And finally, what are some of the next steps to consider when faced with a variant of uncertain significance? So we'll start with some very basic definitions, really just to get us all on the same page. Genetic testing is the use of a laboratory test to examine an individual's DNA for variations. We call these genetic variants, also mutations or genetic change. And the result of a genetic test can be used to confirm or rule out a suspected genetic disease. So this is what we call genetic diagnosis. A genetic diagnosis then may impact management if we have an actionable result. And results may also be used to determine the likelihood of parents passing on a genetic variant to their offspring. We refer them to the recurrence risk. Also, those results may be used to determine the status of affected or unaffected of other family members who may be at risk. We often refer to this as cascade testing. Now, importantly, while we all are carriers for many genetic variants, statements about genetic diagnosis, actionable results, precise recurrence risk, and then the opportunity to test other family members at risk through cascade testing, those are all possible only if we have a positive or a negative test result. Uncertain genetic test results do not inform the diagnosis, the management, or the recurrence risk, and they can't be used to assess other family members. Now, positive or negative genetic test results have what we call clinical utility because they inform diagnosis management, they inform recurrence risk, and can be used to assess other family members. Uncertain genetic test results generally do not have immediate clinical utility, as they do not inform about these aspects. However, understanding an uncertain result helps counseling the patient appropriately and decide on the next steps. Understanding really the uncertain result, not only yourself, but also communicate that information to the families. And as you hopefully will see, is that in some instances, there is actually some clinical utility, even if you have an uncertain result. Now, variants of uncertain significance are inevitable when we analyze large parts of our genome, whether we're looking at large gene panels, an whole exome or whole genome. Just as an example, in an average exome, we may detect between 20 and 40,000 variants, of which 100 to perhaps even 400 may be rare in the population. So it's worth spending a little time on reviewing how the genetic laboratory applies defined criteria to the evaluation of these genetic variants to determine or to predict if they have a functional impact and ultimately whether they are disease-causing. So a genetic variant is really defined as a position in the DNA sequence of our genome, and that is different from the reference genome. And the human reference genome is essentially a DNA sequence database that is put together as a representative of the human species. And so it's really an idealized reference, which means just because a DNA sequence differs from that reference genome does not mean necessarily that it impacts health. In fact, the vast majority of variants or differences from the reference genome are relatively common in various populations. And for a better understanding on how to find the, so to speak, needle in the haystack, meaning the one variant that causes the phenotype or a particular disease, we will contemplate three aspects here. We'll look at the different variant types, we'll look at the variant consequences, and the variant classifications. So let's start with the variant types. So a simplified way of looking quite simplified way of looking at variant types is to distinguish between small variants and large variants. So among small variants, we distinguish between single nucleotide variants, or SNVs, or multinucleotide variants, such as this dinucleotide substitution there that's shown, or then also small insertions or deletions of nucleotides, so-called indels. In contrast, larger structural copy number variants, deletions or duplications, may also be detected by next generation sequencing, or perhaps a chromosome microarray. Now here we will be focusing on smaller variants, although really similar considerations apply to copy number variants when assessing variants of uncertain significance. Just to, for ease of the topic, we'll limit our discussion here to those smaller variants. Now, in a genetic test report, the laboratory typically uses a standardized nomenclature for the DNA change and also the predicted protein change. Now, the most distinguishing feature of that reference genome I mentioned is that so-called RefSeq, you can call it RefSeq record. That's one database, basically the RefSeq that refers to the human reference genome. So that most distinguishing feature is a distinct accession number that has a certain format that begins with 2 characters followed by an underscore. So here we have NM underscore for the protein coding transcript and the NP underscore for the protein that is associated with this particular NM number. Not all labs may give this reference number, or at least not right up front. It might be in the small print somewhere, but it's important because it specifies which protein transcript was considered by the lab when evaluating the variant and when classifying the variant. Now, a gene may have several different transcripts, and the same DNA variant, the same DNA position, may affect a coding region in one transcript and an intron, so a non-coding region, in the other transcript, which is obviously makes a difference in variant interpretation. So bottom line, the RefSeq accession number gives you the context in which the variant is interpreted. Now, if the coding sequence variant is given, this is indicated by a C dot that's shown here. If the genomic reference sequence, for example, is given, we will see a G dot and so on. Now, in this example, the transcript isoform NM underscore 015311.3 was considered, presumably the main transcript of this gene. And then within the coding DNA sequence, the exonic nucleotide position 558 is duplicated. That's what's shown here. An insertion of a single nucleotide, so a duplication of that single nucleotide in an exon is predicted to change the reading frame during a protein translation. And in fact, that's shown below, where the protein sequence corresponding to the reference transcript is shown, and that's indicated by the P dot. So the insertion of that single nucleotide change that codon to result in a substitution from a leucine to a proline at amino acid position 154. But then it also results in a codon frame shift, which inevitably leads to a stop codon at some point, in this case after 100 amino acids. So all of that information, it's a quite rich information, is actually given in this genetic test report. And only in these two terms, so the NM and the NP term here, the nomenclature. And as we see in the next slides, now in this case, I just chose a frameshift variant, but frameshift variants are a variant type. It has a high impact. So it has high impact on protein function, as they either lead to protein truncation or a dosage loss. We call this also haploinsufficiency due to a nonsense-mediated decay. Now let's look at how we predict consequences of genetic variants. So the impact of a genetic variant on protein function depends on its location and the variant type. And here on the top, what you can see is basically the outline, just a schematic outline of a gene with the different kind of regions, and it's like a topography basically of the gene. And what you can see here, it's a little small on unfortunately, I can't see it very well. You can see regulatory regions, you can see the upstream region of the coding region. So this might be a regulatory region, intergenic region, might be a promoter region in the 5' untranslated region of the what we call UTR. And then in orange, we have the exons, which are coding regions proper. And the information of these exons is translated into the protein after transcription. And then we have the introns in between the exons, while changes in the immediate exon-intron border affecting what we call the canonical splice sites are highly likely to have an impact on protein function. Deep intronic changes, or somewhere in the middle of this intron, for example, are much less likely to impact protein function. And more importantly, they're much more difficult to predict. And then going further from left to right, we have the downstream region, including the 3' untranslated region or 3' UTR region. So variants in exons and canonical splice sites have higher impact than variants in non-coding regions, such as the 5' UTRs, introns, or 3' UTRs. In fact, the latter ones, unless it's a well-described variant that causes disease, most of the times the default classification will not rise above a variant of uncertain significance. Now, within exons, truncating variants that introduce new stop codons, such as stop gain variants or frameshift variants, are generally considered to have the highest impact. So those are high impact variants, and depending on whether they occur early in the coding sequence or late, they may lead to either dosage loss or haploinsufficiency due to nonsense-mediated decay, or when they are later in the protein, they may actually lead to a truncated protein. And just on a side note, even though we think of these variants as loss of function variants, if one of those truncating variants actually happens very late in the transcript, this potentially could, and it causes truly a truncated protein. It's actually more difficult to predict the function. It may not necessarily a complete loss of function variant. That might be partial function, or in some instances, this could have a dominant negative effect, perhaps even the gain of function effect. So there is some variability even here in those variants that we generally think have high impact and are predictable. Now, in contrast, the functional impact of missense variants usually affects structure and not the dosage, but it is also more difficult to predict. And we utilize various in silico algorithms that have been developed for exactly that purpose, to predict the functional impact of a missense variant. As a whole, and in contrast to truncating variants, missense variants are generally predicted to have only moderate impact. And then lastly, we talked about it a little bit earlier, exonic variants that don't change. Actually, we didn't talk about this. Those variants in exons that actually don't change the amino acid sequence, we call these synonymous variants. But then also those non-coding variants outside of actual splice regions, such as deep intronic variants upstream or downstream of the coding gene. Those are generally considered to have the lowest impact. Now, this is generally true, but we always check with available tools whether, for example, a synonymous variant may actually have impact on splicing. While a nucleotide change may not result in an amino acid change, could potentially create something like a cryptic splice site, so a new splice site that wasn't there before. And if this new splice site is stronger than the actual splice site at the border between the exon and the intron, it could very well have an impact. It's just something to keep in mind, and just to, kind of illustrate where all this uncertainty comes from. And so another way in looking at this is shown here. On the y-axis, we show the different types of variants, and on the x-axis, we show how well we actually can predict the effect of these variants on the protein function. And in addition, the color tells us whether the effect is more on protein dosage, so this is more dark in dark green, or on protein structure, that's shown in light green. So as you can see, the effect of truncating variants like premature stop codons, frameshift variants, as well as canonical splice variants can most reliably be predicted. And the most common consequence is a dosage change or haploinsufficiency. Now, in contrast, our ability to predict the effect of missense variants on protein function is much less robust, and this is in part because our models, our computer models to predict structural changes, are imperfect. So not surprisingly, among coding variants, reported VUSs are often missense variants. Now, in the following slides, we're going to do a kind of a high-level walkthrough of the steps a laboratory follows when classifying genetic variants. And this process is, as I'm sure many of you are aware, is based on criteria that the American College of Medical Genetics and their Association of Molecular Pathology developed for the interpretation and classification of variants detected on next-generation sequencing. Now, every lab has its own kind of process, individualized process, but really all labs rely on the basis on these recommendations. So let's start with those aspects that are most important from a clinical perspective. I apologize if some of those is very obvious for some of you. So for each variant, the lab will assign one of 5 classification categories. The variant is classified as pathogenic, if there is a strong evidence that it's disease-causing, likely pathogenic if there is at least 90% certainty that the variant is disease-causing. And on the other end of the spectrum, the variant is classified as benign if there's strong evidence that it is not disease-causing, and similarly likely benign if there's evidence to support at least 90% certainty that the variant is not disease-causing. Now, variants of uncertain significance are defined by what they are not. They are neither likely pathogenic or pathogenic, nor are they likely benign, likely benign or benign. From a clinical perspective, it doesn't really matter whether a variant classified as likely classified as likely pathogenic or pathogenic. Both are considered positive results. Similarly, both likely benign and likely and benign variants are considered negative and are typically not reported unless they are reclassified as such, for example. Now, obviously, the result for a variant of uncertain significance is uncertain. However, looking a little more closely at the evidence criteria used in the classification process will help us to better understand the potential significance of a variant that has uncertain significance. And when doing that, we will see that indeed not all variants of uncertain significance are created equal. This slide is getting busier, but we can now focus on the left side. What I want you to see here is that the classification is really the result of a simplified 3-step process. First, we evaluate all the available evidence for a particular variant. And then second, we grade that evidence as very strong, moderate, or supporting for pathogenicity, and then going through a similar process for benign classification. And then third, we add up, or we line up, I guess here, we line up and add up the graded criteria, which puts them in one of the classification bins, pathogenic, likely pathogenic, on using really a predetermined scheme, which we'll briefly talk about a little later. Now, accordingly, a variant of uncertain significance may well have evidence for a disease-causing effect of pathogenicity, just not sufficient evidence. Similarly, a variant of uncertain significance may have evidence to be benign, just not enough to classify it as such. So being able to actually make assertions about the available variant evidence may help you to interpret the VUS, the variant of uncertain significance, in a much more differentiated way and help you with counseling and planning the next steps. So going through these steps for yourself, this may lead you to think of some VUSs as actually favoring benign or other as favoring as pathogenic. So here's the type of data we pull to aid in variant classification. We review population data, computational data, and functional data. And then if available, we may also look at data from other family members to impute whether a variant is de novo or whether it is inherited. And if it is inherited, whether it segregates with the phenotype, or whether or not, if you have two variants, whether it supports biallelic inheritance in autosomal recessive disorders. So let's, for example, if a variant, when we look at population data now, if a variant is absent from large population databases such as NOMAD, that counts as a PM2 criterion, a moderate support for pathogenic classification. If a variant frequency is too high in the population for the disease in consideration, it counts as a BS1 criterion, strong support for a benign classification. If we have actually population data, population studies, case control studies, for example, showing that certain variants are enriched in affected individuals. This may provide a PS4 criterion, strong support for pathogenic classification. So certain alleles from common monogenic disorders, such as alpha-1 antitrypsin deficiency, MTHFR deficiency, for example, but also cystic fibrosis or hearing loss fall into that category. And this classification scheme, based on the population classification, is based on what we know about how population allele frequency affects variant effect size. So in general, when looking at Mendelian disorders, we are considering rare, highly penetrant disorders that are caused by variants that are rare or ultra rare in the population. In addition, low frequency variants with intermediate effects may be found, especially in autosomal recessive disorders, and they may contribute to the clinical variability of these disorders. What I just earlier, the last slide mentioned those variants that are classified through as pathogenic, likely pathogenic through case control studies. So if there's an enrichment in the variant allele frequency in affected ones, those could potentially fall into these. And again, there's examples I mentioned, alpha-1 antitrypsin deficiency, for example, but also some variants in cystic fibrosis or even connexin 26 related sensorineural hearing loss. So that's really the basis for the population-based criteria. Now, computational data can also provide different levels of confidence for pathogenicity. Loss of function prediction in a gene where loss of function is an established disease mechanism provides a PVS1 criterion, very strong support for pathogenicity. An amino acid change identical to a previously established pathogenic variant provides a PS1 criterion, so strong support for pathogenicity. And then lastly, consistent prediction of a damaging effect from different and ideally independent computational model calculations provides a PP3 criterion. So it's a supporting criterion, the lowest confidence level of supporting criteria. And that kind of goes back to what we said earlier. Those algorithms are not perfect. And this also is basically the reflection of the fact that it's not so easy and not so reliable to predict missense variants. Now, testing other family members, as I mentioned, can also be very helpful. A variant not detected in either parent is presumed to be de novo, with the caveat of potential germline mosaicism, of course. And it is considered a strong criterion if biological parentage is confirmed, or moderate criterion if we haven't by testing confirmed biological parents. In the support for pathogenic classification. So these are either PS2 or PM6, respectively. Co-segregation with disease is considered a moderate to strong evidence supporting pathogenicity, depending on how many informative meioses we're actually looking at. And if a variant is detected in trans with a known pathogenic variant for an autosomal recessive disorder, this is also considered a moderate evidence supporting pathogenicity criterion. It's the PM3 criterion. Now, we're not going through quite all of the criteria, and here's a summary table really with the evidence framework for variant classification that was put forward by the ACMG and ANP. The reference is given down below. I'm sure many of you are familiar with that work. But just to orient you on the y-axis are the categories that we just reviewed, and then on the x-axis, the evidence strength for pathogenic and benign classification. And of course, there are a number of limitations, like with every classification framework. For example, some criteria imply subjective judgment. An example, and again, it's a little small here. When you look at the green column, the second to lowest here, the reputable source that provides you with a PP5 criterion, it's quite subjective. Or right below that, the last row in the green column, the patient's phenotype or family history to be highly specific for a gene that provides a PP4 supportive criteria for pathogenicity, also relatively subjective. Now, some criteria could potentially actually be double counted if you don't use appropriate guidance, if the lab doesn't have like a standard procedure to use them. And then there's also no clear guidance on how to combine pathogenic and benign criteria, which is why variants that have both pathogenic and benign criteria that apply often end up as uncertain. Now, here's this earlier slide, part of the earlier slide you saw, and how to line up or combine, the different evidence types and the strength, and how they result in a classification based on a predetermined scheme. And so this is the scheme, the guiding rules, basically, on how to combine these criteria to arrive at a classification. So those are shown in this table. We won't go through in detail, but it serves as a reminder that a variant of uncertain significance may just not have made the cut, may just not have made the threshold to be classified as either benign or pathogenic. And so trying to obtain additional evidence may, in some cases, help you to move the needle away from the uncertain toward a more definite classification. And then just a few words of caution and some additional aspects that may or may not be relevant for you, but I think as a reminder that this is in fact a much more complex. It becomes more complex for various reasons. So first, you need to ask yourself, how well is the particular gene that you're looking at actually established in relation to that particular disease. We call this the validity of gene-disease association. So for example, a variant in a gene with only limited disease association, only limited evidence for disease association, will usually not rise beyond an uncertain classification. Ask yourself, is there genetic heterogeneity, meaning can a particular phenotype be caused by variation in more than just one gene? There are plenty of examples. Noonan syndrome would be one of the examples to think about. So in these cases, you need to check whether all of the genes have been looked at in the particular test you ordered if you suspect that particular syndrome. And then for some genes, different variants in the same gene cause different phenotypes. We call this allelic heterogeneity. An example, there are lots of examples. One example would be SCN1A, loss of function variants. Those cause Dravet syndrome, which is a severe infantile onset of epileptic encephalopathy, highly penetrant. Whereas SCN1A gain of function variants cause familial hemiplegic migraine, very different syndrome. Related to that really is the predicted variant consequence consistent with the known disease mechanism. So if a variant in a gene does not match the mechanism for the disease in consideration, you may not have to worry about that. So to go back to that example, a rare SCN1A gain of function variant detected in an infant with infantile epileptic encephalopathy may not be the answer. So you don't have to worry about that really too much. And then does the genetic test actually detect the variant type associated with the disease in question? We call this the clinical validity of a genetic test. So for example, routine short-read next-generation sequencing typically does not detect repeat expansion disorders or methylation disorders. So be familiar with, and then and I'll have a slide after that just to expand a little bit on that. And then also, I think it's important, to be familiar and really copiously use public databases and curated information sites such as NOMAD, ClinVar, ClinGen, OMIM, Gene Reviews. I didn't put the links in here because you just have to Google them and they come up right away. So I put this very old slide on as a reminder to what I earlier mentioned. Be aware of what test you order to make sure that that test actually is capable and give you that result for this particular disease. And even global tests, including whole exome sequencing and even short reads, whole genome sequencing, may not be able to detect certain type of genetic disorders, which still require specific testing in most routine clinical situations. There's research and there are developments where especially for genome sequencing, might actually give you this information, but this requires usually additional steps that are not necessarily provided yet on routine basis by most laboratories. And if you think they are, make sure that's the case. I often hear from providers, oh yeah, this patient had everything, including whole exome sequencing. We didn't find this. Well, what do you think? What do you think the patient has? Well, we think the patient has myotonic dystrophy. So this you wouldn't detect this by usually by next generation sequencing. So examples are these repeat expansion disorders, methylation disorders, but also SMA, for example. SMA is caused by mutations in a gene that has a very high homology with SMN1 with SMN2, and routine and next generation sequencing is often not able to give you that diagnosis here. And so here we're putting it all together before now moving to a few cases. I mentioned earlier that genetic testing is all about one component that contributes to a genetic diagnosis. The clinical evaluation really remains critical. It may not be as critical anymore to decide on initial genetic testing strategies as the genome first approaches have shown, but the phenotype is still important in result interpretation, including interpretation and weighing of VUSs and deciding on second-tier testing. And the same is true for ancillary laboratory testing, imaging, which are really just part of the extended phenotyping. So now for our case discussions, Jaclyn will join us, and we're going to start kind of with the first case. Jaclyn, do you want to give us a brief introduction to our first case?
Yeah, thanks, Dr. Elias. So I am excited to unpack those concepts Dr. Elias just talked through with some real examples, and hopefully show that we can come to resolution at times for these families facing uncertainty. So this first example is a 9-month-old little one with red cell aplasia, whose provider ordered a multi-gene bone marrow failure panel. What we saw, as you can see, is a variant of uncertain significance in the RPL5 gene, which is associated with Diamond-Blackfan anemia type 6. This is an autosomal dominant condition, and really the patient's phenotype overlapped that with excellent overlap. So Dr. Elias, do you want to talk about why the variant was classified as a VUS based on criteria?
Yeah, and this kind of just repeats now what we talked about it for this particular case. So when you look at this variant is absent in population databases. This buys that variant a PM2 criterion. However, it's not cited in ClinVar or in affected individuals in the literature. So we don't get any additional supporting information from that. So it's not a known pathogenic variant. And then it is a missense variant, and it's a missense variant with multiple lines of computational evidence supporting a deleterious effect. All that buys us is a PP3. So as I mentioned, it's only supporting, at least based on the current guidelines. There might be some changes. There will be some changes ahead, but at this point, that's the case. So also, we only tested the patient. So we don't know whether or not this variant is inherited or it's de novo. So the PM6 or PS2 criteria cannot be applied. And as Jaclyn mentioned, we felt that this has excellent phenotypic overlap, and then really communicating with the provider, further clinical evaluation strongly suggested that the patient had Diamond-Blackfan anemia. Now, the initial then delineation of this classification shows that this variant does not rise beyond the VUS, and I put on the left side kind of this table. Again, when we add up the PM2, PP3, PP4, it's just not sufficient for a likely pathogenic variant. So Jaclyn, what would you recommend as the next step?
Yeah, so as we often do, we try to coordinate parental studies, and in this case, we saw that neither parent carries that variant, so it was de novo in the patient.
So a de novo variant, so do those study results change anything in the variant classification?
Yeah, so the de novo status in this case added a moderate pathogenicity criterion, or a PM6. And by adding the PM6 to the other criteria, we had sufficient evidence criteria for pathogenicity to reclassify the variant from a VUS to a likely pathogenic, which confirmed the clinically suspected diagnosis of Diamond-Blackfan anemia in this little one.
Yeah, so this is an example, I think, where we start out with the VUS, and an additional testing might help us there. So what about the next case?
Yeah, so this was a 4-year-old little girl with bilateral high-frequency hearing loss, a frequent indication that we see. The provider ordered a large hearing loss gene panel, and there were 2 variants identified in the MYO15A gene. And in this case, we were not sure the phase of these variants. And phase, meaning we didn't know whether these variants were on the same copy of the gene or biparental. This gene is associated with an autosomal recessive deafness. And we know that historically most variants in this gene are associated with severe to profound congenital sensorineural hearing loss in consanguineous families. But more recently, a milder phenotype with postlingual, predominantly high-frequency hearing loss has been recognized.
Yeah, and actually, when we look at these variants here now, there's this one variant. The first one is actually a splice variant, splice donor variant. That variant is also absent in population databases. That is a PM2. It's not cited in ClinVar or affected individuals either, so we don't get anything, any supporting criterion here. However, it's at the +2. It shows you that this is actually affects a canonical splice donor site in intron 28 of that myo15A gene, and it's expected and predicted to disrupt RNA splicing. And as I mentioned, canonical splice sites have a high impact, and they're also reliable in terms of the predictability. So this actually gives that contributes a PVS, so a very strong PVS1 criterion, very strong criteria, and PM2 plus PVS1 will give us actually a likely pathogenic variant. On the other hand, the other variant is a missense variant, although a very kind of damaging looking one when you think about substitution of a tyrosine through a cysteine. However, it's present in population databases. NOMAD has it as a frequency of 0.02, which is not very rare, but quite consistent with recessive inheritance. However, we don't get any supporting criterion for this. ClinVar has several citations with classification of uncertain. However, it has not been reported in affected individuals. And then the only criterion we get is that this variant also is predicted by multiple lines of computational algorithm to have a deleterious or damaging effect. So it has a PP3. So PP3 alone only gives us a variant of uncertain significance. So Jaclyn, what did you do in this case?
So no surprise, we did parental testing. And in this case, remember, this condition is associated with a recessive disorder. And so we expect to find variants on each copy of the parental allele. So we found just that. We found that both parents are carriers. And we were able to reclassify the report from uncertain to a likely pathogenic with the diagnosis of myo15A related autosomal recessive deafness type 3.
Great. So maybe let's do a third case then.
Yeah, and this one is a little bit more complicated, but I think we can work our way through it. I would point out clinically, this is not an unusual type of a test report to get back, but we'll unpack it together. So the primary diagnosis for this patient, by the way, another kiddo with hearing loss. Notice he has a homozygous variant in GJB2. So that is consistent with the diagnosis of autosomal recessive GJB2 deafness. And then he has a number of, he has one pathogenic variant in USH2A and then 4 BUSs. So the other diagnosis that we need to consider is Usher syndrome. And the reason why that is important is really because we know Usher syndrome can have other medical features beyond hearing loss. And one of those specifically is vision loss, possible blindness, obvious need for surveillance. So determining whether this child has Usher syndrome in addition to GJB2 related deafness was really important. Abe, would you say could the other variants be dismissed?
Yeah, so I think those other variants we're talking about the LOXH-D1, PCDH15, and the TSPR genes. I think all these, we can pretty confidently dismiss those other 3 variants. Those are all associated with autosomal recessive conditions, and we do not detect a second variant required in trans for a diagnosis. However, in addition, we don't have a strong suspicion for any of these disorders based on their lack of phenotypic overlap with the patient's presentation. So that leaves us with one genetic mystery to be solved, the question on whether this child has not only GJB2 related hearing loss, but also Usher syndrome. So what, Jaclyn, would you suggest we do next?
So big surprise, I would recommend doing parental studies again. And really in this case, we saw that the mother has both of those variants. And so we call that incis. So in this case, we were able to tell the mother that really she's an unaffected carrier of Usher syndrome, just as her son is. The father did not have either of those, as you can see there. So just to kind of recap, genetic testing, starting from start to finish, this is simplified, but hopefully kind of captures some of the concepts we talked about. So we would see a patient with a suspicion of a genetic condition. We would choose an order-appropriate genetic testing. Very importantly, pre-test genetic counseling, especially around this possibility of variants of uncertain significance, is very important. We would test the patient sample, issue a report, review the test report, again, disclosing results, whatever they may be, positive, negative, or uncertain, and then looking toward next steps, which, again, in the case of the US often involves testing family members, parents. That is not always possible. And so we work around different options that way too.
Well, thank you, Jaclyn. And so I think we're towards the end here of our webinar, and just as a reminder, variants of uncertain significance, as I mentioned, they're inevitable consequences of genetic testing. They do not meet criteria for benign or pathogenic, or sometimes the evidence is conflicting. They're not informative for diagnosis, management, or recurrence risk, and really shouldn't be used to assess other family members' risks. They may be reclassified after additional variant information becomes available, and really should be clinically appraised in the context of the phenotype and after you yourself have actually looked at the classification evidence. And they, as in some cases, may be resolved through appropriate family studies. So with this, I close and give it back to Christy.
Great. Thank you both so very much. I really appreciate your time today and this informative presentation. We have quite a few questions that have popped up. So I will go ahead and read off a couple here to you. Some from early on in your presentation. Could you clarify what the numbers represent when you're saying PM2 or BS1?
Yeah, in the interest of time, I didn't go into detail, but for each category, so I mentioned there's very strong, so VS, so the P is for pathogenic support for pathogenicity. Thanks for asking that. The P stands for support criteria for pathogenicity. The B, support criterion for benign. And then there are very strong support, so would be PVS. There is strong support, it would be S. There's moderate support, that would be M, so PM, and then there's just supportive, and that would be P. And then the numbers are basically just the criteria, the different types of supporting criteria. So PM2, for example, would be for the population-based criteria, so not in population databases. De novo would be PM6. So it's the numbers basically define the category where it comes from, and the M and the S, and so the strong, the supportive, the very strong, and the P at the beginning, the B at the beginning, pathogenic versus benign support. Hope that.
Great. Thank you. Thank you for breaking that down. And then just a simple clarification, are all of the coding regions part of the exome?
So no, no, not all coding regions are part of the exome. And the reason for this is that the exome is a capture-based method. Means that before you actually get the DNA, you need to select those coding regions. And there are lots of different platforms available. Each platform has has strength in certain areas of the coding exome and weaknesses. For example, particularly there are certain areas, coding areas that are difficult to capture, low complexity regions, or for example, repeat regions are very difficult. Very high GC rich regions. So regions that are difficult to process in the wet lab process are regions that are not covered. So, and that leads to the fact that really only about, 95 to 97% or so of the coding regions are covered. And even within an existing platform, there's some variability. And that's an important part. And this is, for example, when you do exome sequencing, it's so important. Not all genes are covered equally. If you have, however, if the lab knows that a certain phenotype is expected, it will look and will see, oh, this gene is strongly associated with this phenotype. However, it's not well covered. Let's look at this further. If the lab doesn't know that you're looking at this phenotype, that's gene, there won't be any variance in that gene because it's not covered well, but the lab will also not look at it. So that's where it's so important that there's really ideally a two-way communication between the ordering provider and the lab. And actually, not only the ordering provider, also the receiving provider. As geneticists, we often are on the receiving end. We see that the test was ordered. So don't shy away to call the lab and say, hey, what about, are you aware about this phenotype? Did you look at all of the genes that are associated with it? What's the coverage of your exome for these? So this is a really good question.
Great. And we are just a little bit past the top of the hour. So I have popped the evaluation link in the chat for everybody who would like to claim CME today. And we'll turn off questions at this time, but just want to please everyone join me in thanking Dr. Elias and Jaclyn Haven for their time today and their excellent and informative presentation. And that wraps up our last Time for Genetics webinar. Thank you all for being here. Bye-bye.
Thank you.
Bye-bye.